Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Dr. Paul Valdmanis, who's Associate Professor at the University of Washington. I reached out to Paul recently when I saw a really cool paper that they'd published looking at a enhancer deletion that's protective in African Americans and I guess African ancestry haplotype more broadly in and around the ApoE locus. So we're going to talk about Alzheimer's today, but as I also dug into Paul's background, I just got more and more interested in all the really fascinating research you're doing more generally across neurodegeneration. So this has been a really fun podcast to prep for. We're going to cover a lot of ground. And Paul, I'm really excited to have you here today.
All right, pleasure to be here. Thanks.
Let's start with that paper, and you've done a lot of really interesting work in Alzheimer's genetic risk in general. But I'll try to tee it up from what I understand about maybe the state of the field before your paper. But It's been observed, I think, previously that ApoE44 carriers, so carriers of the risk variant, in general, we know it's a higher risk population, but actually in African Americans, it wouldn't have been as high risk compared to ApoE44 carriers of European ancestry, for example. And it was a little bit of a puzzle, and I think your work helps to shed some light as to why this might be the case. I love if you could correct me if my understanding is off on that, and in general dive into how you thought about this before you kicked off the research.
No, that's exactly correct. It's one of these situations where it's really surprising that there is a differential risk based on population. And the ApoE locus has been known to be such a strong risk factor for Alzheimer's disease for decades now, pretty much. And there's been a lot of kind of research that's gone into this. I should say, stepping back, we've had a lot of interest in genetics research and trying to use new technologies or new methodologies to try to uncover how some of the genetic risks could account for diseases such as amyotrophic lateral sclerosis, Alzheimer's disease, and other approaches. And when I started my lab, it was about 8 years ago, I was really looking for kind of ways to identify variants that could be implicated in disease and using approaches such as long-read sequencing. And so, we know that the risk for individuals of African ancestry at ApoE that are E4 carriers is quite a bit lower than individuals of European, and especially even individuals of East Asian ancestry. And a lot of that work builds off of seminal work from, people like Jeffrey Vance and Jeff Vance at Miami and Elizabeth Blue here at the University of Washington. And it was actually Elizabeth Blue that was giving a presentation, a seminar. And one of the questions that came from that was from Debbie Nickerson saying, there's all these long read genomes that are available. Perhaps we could uncover some of the variants that are on, the same haplotype or the same allele at ApoE, and could that explain, some of this risk? And so that's some of the work that we dove into because we've been accumulating a lot of these long-read sequencing. We've had undertaken a lot of long-read sequencing using various technologies to kind of identify some of these novel genetic risk factors for disease, and that's where we were, really interested to kind of take a look using long-read sequencing approaches to see if there were any variants that were present that might have been missed by conventional short-read sequencing approaches, essentially.
Yeah, interesting. And we'll link the paper in the show notes for people who want to go in detail. But can you talk a little bit about what you found, the degree of protection, and you dove into the underlying biology as well, exactly how and why we hypothesize this to be protective. So I'd love if you could just talk a little bit about that.
That's right. Yeah. So what we were able to uncover is, looking at a larger region around ApoE in, again, some of these long read sequences that have been generated through various resources using Thousand Genomes Project samples. We were able to find a deletion variant that sat very close to ApoE itself. It's only about a kilobase downstream past the third prime UTR. And in aligning some of these reads, we actually, looking at the kind of enhancer tracks, we found, and transcription factor binding tracks, that we found that the deletion actually seems to disrupt a microglial transcription factor named SPI1. And that links a lot of these common themes in Alzheimer's disease biology. We know that SPI1 itself is a GWAS risk for Alzheimer's disease. There are sites scattered across other AD risk genes that SPI1's variants that disrupt SPI1 binding seem to associate with risk. And so, this was a really compelling kind of situation where we thought that the SPI1 disruption would be something that could impact the expression of ApoE. And so that's where we kind of, we had the genetic data, and then we wanted to kind of validate this using functional approaches, because, there's a lot of genetic variation at this locus. There's several genes that are all kind of in tandem that have several variants that also could, have been proposed to explain protection or risk in various populations. And so, this is one of the ones where we wanted to kind of, exactly take that sequence and clone it out. And so from that, we were able to identify, that this variant seemed to disrupt an enhancer site that normally would repress ApoE, but only in microglial cell lines, not in astrocytes or neuronal cell lines to the same extent. And when we looked at various databases, both from the Alzheimer's disease sequencing project, as well as the really powerful All of Us resource, we found that the variant from a genetic standpoint was about as protective as the ApoE2 allele if you correct for ApoE4 status. So meaning that, the variant on its own, is not, it doesn't have a drastic kind of reduction in protection for Alzheimer's disease, but that's because it's only on individuals with the ApoE4 background. And so, if you take into account the ApoE structure, that's where this protection dropped nearly in half in terms of the risk for Alzheimer's disease. So this is, one of the situations where, looking at population-specific databases and samples can really uncover areas of the genome and variants that previously were underappreciated for playing a role in expression in disease.
Yeah, it's fast. So you're saying the protective quality is only or primarily in E4 homozygotes, is that right?
That's correct. Yeah, we see it in up to 75% of individuals with an E4 allele on the African ancestry, and we see rare, extremely rare instances where you might find the deletion in an E3 allele and never on the E2 allele. So that's something we're really interested in. Is, is there some kind of like synergistic or combined protective effect that now we can learn once we've identified this new variant.
Oh, interesting. So they are, because they're so close together, they're really on the same haplotype. So most in the paper you have breakdown of different populations, but people are getting the protective allele and the E4. It's the same haplotype basically in the majority of cases. That's really interesting. Yeah.
Yeah, that's the beauty of some of the longer read sequencing. Is we don't have to work on just kind of like inferences based on the correlation between steps. We have the actual landscape across each allele, in each haplotype to identify how many people have this protective variant as well as the E4 allele and anything else on that same region.
Now, if you were able to go in and engineer the change by knocking out the enhancer with CRISPR or something like that, what would the hypothesis be in all backgrounds? Do you think it would be protective, broadly speaking?
That's the hope, and that's the excitement of some of the future work that would stem from this. Is what is the exact cell type that's implicated? Is it only microglia? Is it a subset of microglia? Are they microglia that are responding to certain stimuli? There's a really kind of a wealth of possibility of how this could play out in terms of functional impact, and disrupting this enhancer. You don't need to have necessarily the deletion. There could be other alternative mechanisms you could contemplate.
Yeah, wow, that's super cool. Is there? I want to move on to some of your research more broadly, but is there anything else on this paper that you want to call out that was particularly interesting or surprising? You have so much interesting stuff I want to cover ground, but I want to do this paper justice as well.
Justice. Yeah, I think the only other surprising feature was that it seems like a lot of the regulation also takes place through an adjacent gene called ApoC1.
Right.
And so it seems like it's an indirect mechanism that the deletions affects ApoC1 expression, which in turn then helps regulate ApoE. And so this was certainly something that has been described in the literature previously in other kind of papers that have looked at, the interplay between some of these genes as a focus. And so that was something that, again, it was unexpected, and it threw us for a loop for a little bit, but, it's something that we're seeing from this data. There's one other thing that I wanted to say that was also quite surprising, was that when we used the All of Us database, so this is like hundreds of thousands of genomes that have been sequenced across diverse populations in the United States, coupled with electronic health records, was that we could see the really defined risk for not only Alzheimer's disease with the ApoE4 variant, but also hyperlipidemia and other lipid traits that had very significant associations. And the deletion variant that we saw reduced the risk for Alzheimer's disease, but not the lipid traits. And then we use that information to identify other variants kind of in that ApoE to ApoC1 intergenic region that had only seemed to have roles in risk for Alzheimer's and dementia-related traits, and not some of these lipid traits. So there's a lot more at this locus, and a lot of these, biobank scale approaches can help tease out some of these and potentially uncouple some of the risks for neurological phenotypes as well as lipid-related traits.
Yeah, really interesting. You've also done, staying in Alzheimer's world, some really interesting work on some of the rarer genetic subtypes, PSEN1, PSEN2, both on the genetic level, but also long-read sequencing of RNA. Can you talk a little bit about what are these, rare, even rarer genetic subtypes? I don't even know if I would call them genetic subtypes, or are they just a different, something different entirely? What are they teaching us about Alzheimer's?
I mean, and what we know from Alzheimer's disease, it primarily initially was from some of the initial gene discoveries that were made, including from folks at the University of Washington that were able to identify PSEN2 variants in Alzheimer's disease. And when we were looking at families with Alzheimer's disease, we were noticing there were some variants that were coming up, including one particular variant of PSEN2 that had a 2 base pair deletion. And almost all of the variants at PSEN1 and PSEN2 are these kind of dominant gain of function variants that seem to disrupt the gamma secretase complex and impact amyloid processing. And so if there was a deletion, this might be a loss of function, maybe one of the first, it would change kind of the perception of how these variants lead to disease. But when we were able to kind of collect tissue samples from some of the individuals with this deletion, we actually found that there was this intronic retention product that actually added 77 base pairs adjacent to the next exon. And so when you added these 77 base pairs minus the 2 base pair deletion, it was suddenly 75 base pairs. And then this was divisible by 3 with no stop codons in this area. So it suddenly restored the reading frame. And it was kind of the exception that seemed to prove the rule that, you have to have these types of new variants that are showing, or gain of function variants that are showing up in this. And so that was kind of the impetus for some of these projects that we were looking at, again, leveraging our experience and our excitement in kind of what long-read sequencing can show us, was that we thought, okay, well, we saw this the combination using short-read approaches, but is the full transcript being expressed? And is there, something that's taking place that also could help us understand, some of the regulation going on at this region? And so for that, we did what's called a kind of a pull down for PSEN1 and PSEN2 for individuals with both familial pathogenic variants in PSEN1 or PSEN2, and then sporadic Alzheimer's disease, sporadic, late onset cases that don't have a defined genetic cause, and that accounts for over 95 to up to 99% of Alzheimer's cases. And then we also looked at controls. And when we performed high throughput long read sequencing targeted at these loci, what we found was quite surprising. First, we did find that 2 base pair deletion coupled with the intron retention product, but we also saw this in sporadic cases. And we saw another feature at PSEN2, what's called a cryptic exon inclusion. And so this includes a portion of DNA that normally should be in the intron, and it was part of the exonic sequence, but it was only present in individuals with sporadic Alzheimer's disease or highly enriched individuals with sporadic disease, not in controls and not in individuals with PSEN1 or PSEN2 variants, and then that already have that kind of genetic hit that would cause their disease. And so, again, going back to my kind of experience with ALS research, we know cryptic exons and alternative splicing, FACN2 and ARC13A are well-defined roles and biomarkers almost now for TDP43 pathology and roles in ALS. So to find something equivalent in Alzheimer's disease is quite exciting, especially at one of the key genes that is implicated in Alzheimer's disease.
Yeah, what connects you've done a ton of work in ALS, you've done a ton of work in Alzheimer's. Obviously they're connected via neurodegeneration, but I think in your case it's something a little bit deeper beyond that, because you're doing work across circular RNA and cryptic splicing. If there doesn't have to be a common thread behind every lab, but what's the thing that connects at least these 2 things in your mind?
I think in many ways it's really, a lot of these neurodegenerative diseases are fascinating, how you can kind of live your entire life without kind of any type of phenotypic consequence, and then suddenly have a drastic decline in whether it's cognition or motor function or so forth. My PhD training was at McGill University. There was, Guy Lowe had collected a lot of families in the Quebec area, but also around Canada. And there was, it was really exciting time to be part of the ALS field and, using some of these well-curated families to try to resolve some of the early familial genes implicated in ALS. And so I have always been passionate about ALS research. And we've used some of these information and some of the information when we were really missing some of the genetic causes for familial ALS. That was where we was realizing we were kind of only looking under the lamppost, only under the areas of the genome that were sequenced down, that were available. And again, some of the long read sequencing that's been able, and the telomere genome approaches that have been able to kind of uncover areas of the genome that were previously intractable with sequencing, kind of inspired me to kind of use, again, some of these long read sequencing to look for tandem repeat expansions. And so one of the ones that we found was in an intron of a gene a gene called WDR7 in ALS. But as well, looking at these, we always wanted to have, again, a control population. And so, at the University of Washington, there's some wonderful resources in an Alzheimer's disease research center where we can actually kind of then connect out, if you have something that's accounted for ALS, it presumably should not also cause Alzheimer's disease, even though there are, TDP43 pathology between the two diseases and so forth. But that's where I got really excited about, the ability to kind of identify things that, may be implicated in ALS that are not in Alzheimer's disease and vice versa. So we were able to kind of toggle back and forth between the two diseases to really hone in on something that's disease specific and not just a function of normative aging and other features that are involved in decline over the lifespan.
When you think about therapies for neurodegeneration in general, do you see a future where there are some, master keys in a way to slow neurodegeneration more broadly across a wide spectrum, or do you more imagine a world where we're more and more targeted with genetic subtypes, and you do a lot of work in gene therapies of different varieties? So I'm interested to just have your perspective on how you see that evolving.
Yeah, I feel it's in some degrees it could be the latter, the situation where there's targeted approaches for in a case by case basis as we understand some of the genetic variants that are implicated, or some of these potentially a repeat expansion that you could target to prevent kind of some of the decline. Some of the beauty of some of the gene therapy approaches is that they are modular. You can swap out, the region of interest to target a particular gene. And so learning about kind of the approach more broadly is quite valuable, because then you kind of, if you have a new subtype of a disease or a new neurological condition that seemed to pop up, on a yearly basis when I was doing my training, it's one of these situations where you can kind of just take the sequence and change it based on the condition and based on the neurodegenerative disease that's implicated. I do think that.
Yeah, and I mean, in these complex neurodegenerative diseases, how do you think about which mechanism to go after? Like, I think about C9 or 72 in ALS, or there's depending on when you intervene, it is kind of a cascade to think about here, right? And how do you think about breaking the problem down, again, with therapeutics in mind, for some of these examples where we have a few different competing mechanisms or a cascade of different mechanisms?
No, and yeah, you bring up a good point, is that a lot of these situations, you really have to go in early and target some of these neurodegenerative conditions prior to some of the symptom onset, or when the time you've lost the neurons, you're maybe too late in the disease process. And so I'm really optimistic with a lot of the the gene therapy approaches, again, starting with things like SOD1 and tofersen, a therapy that can, an antisense therapy that can target this molecule early in disease stage, and how that can protect against some of the neurodegenerative decline. And so really understanding, those have worked really well in familial SOD1, maybe not so much yet in C9 or 72, but understanding the approaches, leveraging again some of the familial forms of ALS and Alzheimer's disease to really kind of target some of the implicated genes where we know that there's a high probability that individuals are going to develop neurodegeneration, and then leveraging that information to kind of go at the larger population as a whole. Are there particular genetic variants or subtypes or predisposition, the PSEN2 cryptic exon that I was mentioning, is this accumulating as a function of aging, and is this something that we can target at an early stage prior to some of the phenotypic decline?
Yeah, if we think about a disease like ALS, and this is maybe just another way of me asking the same question about whether there's a way to treat really far upstream, or whether it needs to be genetically targeted in some way. I'll break my question down this way. From a biological perspective, is there a way that if we were able to run a clinical trial, there's plausibly a way to treat early enough that you could get all or the vast majority of cases early enough that you could prevent disease? Or is there enough differences in the heterogeneity in the population that actually a C9 case, a SOD1, a FUS, you're hitting such fundamentally different pathways that you probably need different tools in the toolkit? And I want to ask the same question about Alzheimer's as well, and whether it's same or different. And I guess I'm getting at whether these are the same underlying disease or not, and what the genetics in your work tells us about that.
Yeah, and I think for ALS, it's the condition is more homogeneous in terms of the phenotypic presentation, and especially even from a genetic standpoint or pathological standpoint, over 97% of individuals with ALS are going to have TDP43 inclusions. And so this is an approach that, if you developed a therapy, could be applicable for a broad swath of the population. And so, again, one of these things, looking at the kind of the converging features that could be implicated, I think a lot of the therapies that may be impacting some of the key components that are taking place, whether it's TDP43 aggregation, nuclear to cytoplasmic dislocalization, or other features, could work for the broad majority, and then the subset of individuals with pathogenic variants may need a separate approach. And so it is possible, again, learning from some of the information from familial forms of disease to kind of use a therapy for a broader population.
And what do you think about Alzheimer's? Is it a different story there?
Alzheimer's is also, had limited success with therapies for the longest time, and it's only more recently with some of the anti-amyloid antibodies and therapies that we've suddenly had some clinical success. And so that has at least provided a considerable degree of optimism that there are ways that we can kind of at least clear amyloid and figure out how that can work to help prevent some of the cognitive decline, but there's clearly a missing component. And so, it is one of these things that may be multifactorial. We may need to take experiences from the cancer field or other indications and, use a combination of molecules. But I think that we're kind of honing in, and it's an exciting time in the Alzheimer's space that, that now we're actually have something to start to work with. And whether, there is a particular target that is going to help be applicable for the whole population is unclear. I think there's, again, a lot of different subtypes of Alzheimer's disease that could have different components that are necessary. But certainly understanding, again, amyloid accumulation, the connection with tau, and how tau tangles are implicated in disease is also critical for some of these therapeutics to be successful.
Yeah. You mentioned oncology and that we can learn from oncology there. You actually have a oncology work stream in your portfolio as well. So you've been working on microRNA and liver cancer. What's the origin of that work, and what do you think neurodegeneration can learn from the advances that have been made in cancer more broadly?
Yeah, that's a good question. And a lot of that stemmed from kind of the early stages in the gene therapy field, where delivery to the brain was challenging and was nascent. And this a lot of the stemmed from when I was contemplating what to do for postdoctoral training, and a lot of the families that I had worked with that had neurodegenerative diseases. We'd go out to remote communities in Quebec and collect, many generations of families all at once. Neurologists would be there to perform neurological assessments. We'd have nurses to collect blood. I'd help with putting together the pedigrees. And in communication with some of these families, it really inspired me to find ways for gene therapy correction for some of these diseases. And so that motivated me to kind of really dive into the gene therapy space. And again, like I mentioned, the ability to target the brain was limited. Ability to transduce hepatocytes was phenomenal. You can hit about almost 100% of hepatocytes with adeno-associated viral vectors. And so it really kind of inspired me to kind of take a bit of a detour in my career, understand how to safely and effectively deliver gene therapy to an organ, the liver, that had well-established techniques and technologies, and then leverage that information to hit the brain as the tools and technologies developed. And so when we were doing this, there was this big concern that there was a lot of off-target toxicity associated with short hairpin RNA delivery. And when you deliver adeno-associated viral vectors to the liver, there was non-specific toxicities dependent on the sequence. And so we knew that if you delivered too much short hairpin RNA for RNAi-mediated knockdown of a gene, it would be bad for the mouse, even lethal. But we didn't know kind of like the mechanism or the buffering or the maximum dose that we provide. And at the time, a lot of small RNA sequencing tools had recently been developed. And so we leverage, again, the new technology, and in this case, small RNA sequencing, to really quantify, to what extent could you knock down a therapeutic gene of interest in the liver before you overwhelmed the existing microRNA machinery. And we found this really unique mechanism associated with miR-122 competition. So this is the microRNA that's accounts for 70% of liver microRNAs. And the more that you delivered these short hairpin RNAs, and the higher the abundance and the expression, the more this would compete with miR-122. Acting as a buffering microRNA. And not only was it competing out, it's actually competing out with a very specific newly synthesized isoform of this microRNA. So we really got into the weeds of how these microRNAs were being competed out by exogenous delivery, and learning mechanisms to kind of, if you could prevent ways to overwhelm miR-122 expression, you could safely and effectively deliver some of these therapeutics. And then the connection,
that's really interesting. So that I didn't appreciate this though. So like more than half of the microRNAs in the liver are this specific, and it needs to be that way to maintain its homeostasis, basically.
Yeah, and then that's the connection with cancer, that if you completely eliminate miR-122, either at birth or later, using some of these overexpression approaches, that a lot of the mice would then develop hepatocellular carcinoma. And we found that there was a particular imprinted locus that was upregulated that had a lot of cancer-driving microRNAs that miR-122 appears to be suppressing. And so you have this embryonic switch between rapid embryo growth and these microRNAs. And it's a DLK1 diode3 locus that's in humans and mice. And if that locus becomes reactivated based on loss of miR-122, that's where you started developing liver cancers in the mice.
Wow, that's fascinating. And what's your perspective on gene therapy to the brain today? There's been a lot of progress, but maybe slower than we would have hoped as well for the field. I'd love to have your perspective on where you see that going.
I think that we're learning a lot of lessons from natural ways that the brain can be targeted, and so there's been a lot of really exciting ways that vectors have been designed that can whether cross the blood-brain barrier or hit particular receptors, like transferrin receptors, that can transduce wide amounts of the brain through some of these vector approaches. And so, again, using some of these tools, you're able to get a very high level of expression now. Previously, quite a bit higher than previously with available. But again, now we have the safety considerations to kind of keep in mind as well. So as you're able to transduce very high levels of the brain, maybe there are safety considerations depending on what cells of interest are hitting. And, there's been efforts to de-target things like DRGs, and other kind of cell types that are involved in like kind of sensory issues that can be overwhelmed using these approaches. But certainly there are, a lot of the safety concerns need to be considered when treating some of these neurodegenerative conditions.
It seems like you're doing a ton of interesting long read work. You're looking at variable number tandem repeats. You're looking at RNA in different tissues. I want to maybe use the phrase you used earlier about looking where the light is. And it seems like you're making a bet right now that there's a lot we can learn from leaning into RNA or long read sequencing rather in a lot of different contexts. Is that right? And where do you think the most interesting places are going to be for long reads?
Yeah, that's exactly right. We're really optimistic about, using long read sequencing to capture some of these areas of the genome that were previously very challenging to sequence. And one of the variable number tandem repeats that I was mentioning, this gene WDR7, kind of really inspired some of this work, where it's a 69 or 68 base pair repeat that can be copied between 1 and 86 times in the human genome. And it folds into actually a microRNA-like structure as well. So there's kind of connections with some of the microRNA that we were doing as well. And what we found was when we could do targeted, again, long read sequencing across this repeat, was that there was a very structured organization to the repeat and a very defined mechanism of expansion that we could identify. And so for that, we were able to deduce that there actually was kind of a directionality of how this repeat expands. There's 6 out of the 69 base pairs were divergent, and each of those could, if we could kind of color code those, we could actually see how the repeat was expanding throughout worldwide populations. And in genetics, we work a lot with phylogenies, and here we could actually see kind of branching off of the repeat as they go from individuals of African ancestry throughout the world. And that got us really inspired to look at some of these other repeats, how they expand and how they diverge, and how they could lead to population-specific expansions that could contribute to disease that may not be appreciated if you only focus on European populations. And so I think that there's really this idea that there could be these expansions that are occurring, throughout the world in population-specific events that could predispose to neurological conditions or other conditions that really have not been appreciated previously. And so combining, again, diverse cohorts of samples with long-read sequencing and capture of the internal structure of some of these tandem repeats throughout the genome, I think it's going to be kind of the next wave of genetic discoveries. That for some of these diseases, really where the genetic culprit hasn't been identified, there's going to be a lot of these examples coming out that there are some of these tandem repeat expansions that just weren't picked up because we didn't have the technology, and now that we do, now there are long-read sequencing and in thousands of individuals, we're really going to be able to capture some of this information that we were totally missing in previous approaches.
Is it starting to happen en masse with some of the scaled biobanks like UK Biobank or all of us, or is there a timeline on long read sequencing across everyone? It has seemed like the big biobanks have gone exome sequencing, then short read whole genome sequencing, but that that next chapter of, okay, now we're going to long read sequence all 500,000 or million. I don't know if I've heard any timelines or commitments to that. Do you know if that's
we're more in the thousands to 10,000 samples, but I think that this idea is really exciting, and I think that this is kind of like the next wave, I think, of genetic discovery is when you get that critical number of individuals, especially if they're combined with electronic health records. We can find instances where there may be an expansion event, and we don't know necessarily even what the phenotype might be, but you can go back to kind of connect that. That's some of the work that our lab is doing right now as well, is where we're doing something called PWAS approach, genome-wide association studies, where we've actually identified some of these expansions that seem to be disproportionately large in certain individuals, and then connecting those back to, okay, what were some of the potential phenotypic traits that might have been associated with some of these expansion events. And so I think we're going to see a lot more of this in the near future. I'm really excited about it.
Yeah, really cool. Last of the recent research that I want to ask you about is some of your ancient DNA work. You were part of a really interesting paper looking at a gene called MUC19 that was part of a Neanderthal, Denisovan, modern human introgression. Could you talk a little bit about how the story behind that? Well, a little bit about what it does and how you learned it, but how did you get involved in the research as well?
Yeah. And again, that was a really fascinating collaboration with Amelia Huerta Sanchez at Brown University. And then she's done some seminal work, really trying to identify regions of the genome that are under adaptive integration or potentially positive selection from Neanderthal and Denisovan genomes. And so she actually gave a seminar here at the University of Washington in the Department of Genome Sciences. And we had some conversations after that, because they had honed in on a region where they had a very strong signature of adaptive integration, in a place where they had both Denisovan and Neanderthal DNA that intermixed and had seemed to account for a lot of positive selection in individuals of Mexican American ancestry and indigenous American ancestry. And they knew that there was like this signal here, but they hadn't honed in on the gene. And our experience with tandem repeats actually kind of allowed us to kind of look at this from a kind of a repeat expansion lens. And we knew, okay, but these mucin genes, there's about 20 or 22 of them throughout the genome, and their defining feature is a really long exon with a tandem repeat and a coding exon. And this tandem repeat is then post-translationally modified, undergoes post-translational modification, glycosylated, that helps contribute to its mucin-like structure. And so when we heard that this was, at this region, we thought, okay, well, let's use some of our tools to quantify the length of these repeats across some of the thousand genomes populations. And so we take a look. We have a tool called Inventor, INVNTR, where we are able to kind of capture a lot of the long reads that are available. And then identify the length. And so with this MUC19 repeat, it's a 30 base pair repeat. And across populations, it's, between 8,000 and 12,000 base pairs long. It's a really long coding exon, like hundreds of copies of this repeat. But in the individuals of American ancestry, we actually saw that it was more than doubled to 24,000 base pairs. And from the long-read sequencing, we'd actually to geek out and find that it was exactly 3,171 base pairs that was copied 5 times instead of once. And again, this is it's not divisible by 30, but 3 of the repeat copies were 27 base pairs instead of 30. So again, it was this divisible by 3 coding exon kind of event. Wow. And so we were blinded. We sent the data to Emilio Fuertes-Sanchez, and they had data to identify which individuals had denisovan-like integration, and there was a perfect concordance, nearly perfect match between the individuals that had this expansion event and individuals that had denisovan ancestry. So our hypothesis is that this expansion event occurred on the backdrop of this denisovan integration, and that's what's explaining a lot of this positive selection and integration signal. Wow. But again, it's something that occurs only in individuals of American ancestry. So we wouldn't have picked up on this if we'd looked only in European samples or other populations.
And so this was happening presumably during the migration over the Beringian land bridge into the Americas, basically at some point in history. How long ago are we talking?
Yeah, and we see signatures of intermediate steps actually in East Asian samples. So we can see this is again, there's 5 copies of this structural duplication event. So there's 3 copies in some individuals. In some places we could see 5 copies, but I think a contraction at other events. So we can actually really trace kind of some of the origins of this expansion event. And it really may be that this expansion event could have happened earlier, but there was something that was protective as humans migrated to kind of across the Bering Strait that helped protect against some of the new climates and new situations that the people were observing and experiencing in the Americas.
Yeah, interesting. I find the ancient genomes amazing. I can't get enough of these results. They're just it's just a fascinating window into something you never would have imagined we could learn about, that we just have a window into time. It's really cool. Mm-hmm. I want to just ask, as we wrap up here, about what's next for you. You've got a ton of interesting work going on. If you think about the next couple of years and what you're most excited about, I'd love if you could just leave the audience with a few perspectives on what are the big projects on your mind.
Yeah, we really are excited again about tandem repeat expansions and looking across populations and some of the long-read genomes that are available. I think that there's, picking out some of the areas that where some of these repeats are expanding to a great extent, and especially in a population-specific manner, and then connecting those to phenotypic traits, whether they are ALS, Alzheimer's disease, or other features that are maybe occurring earlier that could explain, why some of these repeats may have expanded. They may have been protective in certain contexts that now, as the human population ages, now are negatively impacting, neurodegenerative disease, essentially. So, looking for these connections between early events and how these expansions occur, and then some of the phenotypes that are implicated, always with the lens of how this is playing out and shaping out in neurodegenerative disease.
Yeah, amazing. Well, Paul, thank you. This has been a super amazing conversation. I've learned a ton in the 45 minutes we've had. So thank you for taking the time.
Appreciate it. Thank you so much.
Thanks, everybody, for listening. And as always, we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate it if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.